here? Yeah. Uh, can you tell them to project your presentation so that he is keynote speaker for Dr. S. Natarajan Award. His topic is novel genotype phenotype correlation and exploring potentials for gene therapy in Stargardt's disease. Dr. Purn Chandra.
Thank you so much. Thank you, AIOS, for giving me this opportunity.
Yeah, the marks will not be there for these two keynote speakers. You don't mind, I think. Okay.
So I presented this work last year. Over 1 year, there are some updates also which I presented recently in VRSA also. I thought I would take this opportunity to give an update on what was happening last year and what is something new which we have found. So as we all know, Stargardt disease is originally described by Carl Stargardt's most common juvenile macular degeneration due to ABCA4 mutation, and 95% of Stargardts are due to ABCA4 mutation. The genetics of ABCA4 revolves around a transport protein which is involved in accelerating the removal of all trans-retinaldehyde. So this is the final pathway. Whenever there is a defective ABCA4 mechanism, there will be excess accumulation of lipofuscin and other byproducts, which the RP cells and the other cells fails to digest, resulting in apoptosis and disease manifestation. Purpose of our study was to report novel phenotypic pattern genotypes in Indian cohort. This was an observational study. So this was the roadmap for our study: identifying families with Stargardt disease, identify and isolate the mutation, studying this mutation in cell lines, how to repair this mutation, and how to monitor the repair. So our study cohort had 39 families, so among which we used whole exome sequencing to identify and isolate the mutation. So all patients, as I told, underwent detailed examination and electrophysiological tests were done wherever it was required. So coming to the results, the age ranged between 9 to 64 years with a mean of 24 years. The consanguinity was present in 28 out of 39 families, that is almost 75%. So a spectrum of fundus abnormalities was detected. This was a 28-year-old male with a typical Stargardt-like phenotype. This patient had homozygous mutation ABCA4 gene. The second one is a 30-year-old female, also had Stargardt's ABCA4 mutation, but there was only atrophic macular changes and no flex at all. So this was one more 46-year-old male with an extensive degeneration involving macula and also the mid-periphery, and along with that there was kind of an outer retinal cavitations in one eye, which possibly could be related to an associated myopia. So this was an interesting family where the daughter, being younger, was symptomatic with the Stargardt disease, fundus tissue of foveal atrophy. So this patient had compound heterozygous mutation ABCA4, that is 2 mutations and a different location of the same gene. Mother was asymptomatic, was homozygous for one of those mutations. When we examined the mother, fundus was absolutely normal. However, the autofluorescence and OCT did show some perifoveal changes. So this is the typical example for same disease, same gene, same family, but depending on the inheritance pattern and the mutation pattern, the disease severity and progression could be different. So this was one more case of 43-year-old female was diagnosed as Stargardt's elsewhere. So she had a family history of father having some retinal problem. When we screened, the father had atrophic macular changes and maybe resolving flex in the perimacular regions. So we ended up finding out a gene called SERPINF1, which has never been reported in the literature. But the interesting fact was the children who were totally asymptomatic were also carrying the same mutation. So when you screen the children, the fundus was absolutely normal, OCT was near normal. But if you look at the autofluorescence, definitely there is an abnormal autofluorescence in the macular region. So this is the OCT and autofluorescence across 3 generations with increasing grade of severity. So most of the mutations in our cohort was missense mutation. Among these, 7 were novel mutations. We wanted to do an in vitro study in this thing, which is a recent update. So to study the effect of this mutation, what we used was a HAK cell lines, and we used Western blot analysis for the protein function. So to make it simple for understanding, this is the control. So these are the different mutations of ABCA4. The darker the band, better is the protein function. So for example, the proband is a 13-year-old male with a relatively good vision of 6/12. This patient has V type of mutation. That means relatively better protein function. So the phenotype is also less severe as compared to the 9-year-old who is 4 years younger, but the disease is more severe because this patient has a Q type of mutation. All are same ABCA4 mutation, but depending on the protein expression, severity could be different. So the ERG was also telling the same story. In the first patient, fulfilled ERG is almost near normal. In the second, there is a diffuse dysfunction indicating a rod cone dystrophy type of phenotype. So, on how to repair this mutation? So, there are some challenges when it comes to larger genes like ABCA4, USHA2, because this cannot be loaded into the single vectors because the cargo capacity is limited. So, we came up with a novel dual vector strategy where we split the gene into two halves, loaded into the separate variants, and once they were co-infected together, they reach the target site where they are intended to produce the expected proteins. So these are the histological studies which we are doing last time. Now we have come to a stage where we are doing the live ERG in the live animals. So if you look at the control, the intact vector and the animals which received our dual vector, the staining pattern is almost comparable. So our vectors are reaching the intended site.
I have 1 more minute for you.
Yeah. So what was known as this is how a Stargardt disease looked like with atrophic macula and the typical flex and outer retinal loss and the OCT. What our study adds is we demonstrated genetic sequencing and screening of Stargardts in Indian families, presence of new genetic and phenotypic variations, and experimental models for pathogenicity of mutation, and animal models to study the dual vector for gene therapy of Stargardt disease. These are our ongoing projects. So this is the wild type mice, that means the mice with a normal ABCA4 function. This is the mice with ABCA4 knockout. So this is a recent update. If you look at the lipofuscin pattern, this excess lipofuscin accumulation. So once we give the injection, the lipofuscin will start dissolving. So we managed to do ERG in these mice. If you look at the mice which received our dual vector as compared to the control and non-injected vector, definitely there is improvement in both A wave and B wave amplitude, indicating both restoration of inner and outer retinal integrity. So our future plan is obviously to start human trial. Hopefully next AIOS I will be presenting that data. Thank you so much.